When you want.
Well, good afternoon, everybody. It's a pleasure for us to be here. I am Tania Cemeli, and he's Isidre Felip, and we work at the Institut de Recerca Biomedical of Lleida. Isidre is a member of the oncologic pathology group, where the investigation is focused basically on cancer, on endometrial cancer and melanoma. And I'm a member of the cell cycle group. Cell cycle means cell division, so we study molecules that are involved in the division of the cells and other functions that they can have. Well, today we are here to explain an interdisciplinary work that it is a collaboration of the both groups, that it's how alterations in cell cycle can promote tumor metastasis. Okay.
Sorry.
So I will introduce a little bit this work. So first of all, I would like to explain you what is a metastasis. We have a tumor in the top of the image here. And there are a lot of cells that they are growing and they are dividing very fast. But there are some of these cells that in this tumor that have the ability to cross the cell matrix and then to intravasate into a blood vessel. Once they are there, they are transported by the blood and to different parts of the body. And when they feel comfortable somewhere in other tissue or when they receive other signals, then they extravasate to a new tissue and start growing a new tumor there. So that is a metastasis from the first tumor. Then here we have two proteins that are important in this work. One of them is cyclin D1, that is an important regulator of the cell cycle. And we have CDK4 that makes a complex with the protein, and then it starts to phosphorylate different substrates. Phosphorylation is a modification that activates different proteins. So in this case, in our work, cyclin D1 and CDK4 phosphorylates a protein called paxillin. This protein is important because when it is not activated, when it is not phosphorylated, it lets the cell to be attached at the cell matrix, so the cell doesn't move. When this protein is phosphorylated by the complex cyclin D1 CDK4, then the protein has the ability to walk, to migrate. So, due to the fact that cyclin D1 is an oncogene and it's overexpressed in a lot of tumors, overexpression is that we have a lot of molecules of this, a lot of quantity of this protein in that cell. So maybe the cells that have high levels of cyclin D1 will phosphorylate more paxillin, will activate more paxillin, and then this would give the cell the ability to migrate. So the cell would be able to intravasate into the blood vessel, then to go somewhere in the body, and then when it feels comfortable or it receives some signal, then the cell would extravasate into a new tissue and it would grow a new tumor there. So here is our hypothesis in the simplest way that we can explain. So Isidre will talk you now a little bit of our results.
Thank you, Tania. So we had this hypothesis. So what you do when you have an hypothesis? You do experiments, right? To confirm the statement or not. So first we start by in vitro experiments. We use these cells there in blackie, are blah, 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 which are from red prostate tumor. Okay, so these are cells that are known to be really metastatic. Okay, so we create this setting here, at the down part, left down part here, to simulate the blood vessel. So we put cells here in the down part with the media, then we put an artificial matrix called matrigel, or we can make it, whatever, to simulate the endothelia, which is the wall of the blood vessel. Okay, so the cells that have migration potential or metastatic potential are able to go from this part to the upper part where there is media with an attractant, a chemoattractant for the cells, okay? So then we count how many cells are able to do that or no, okay? So we have our control here, which is our 100% migration cells, okay? Then what we do is this column here, which is down-regulate the protein cyclin D1. That means that we have less levels of this protein, okay? And we see that the migration potential of these cells has gone down. Then we did other things, but the other important one is this one here, where we have the downregulation of the protein cyclin D1, but at the same cells we have an hyperactive form of paxillin. Hyperactive form means that paxillin is always working, okay? And what we saw here? We saw that this migration potential, or the metastatic potential, is recovered. Okay, so we have these kind of cells. When we add an hyperactive form of paxillin, they are metastatic again. So after in vitro results, because I usually work with animals, we do in vivo experiments. Okay, so we use different kind of mice. We need to use immunodeficient mice to inject our cancer cells to see if they are able to do metastasis in the animals or not. Okay. Why immunodeficient mice? Sorry. Because if I inject some of you, or I inject myself, some cancer cells, they won't grow, because your immune system will kill them, okay? Because they detect like a foreign thing. That's why we use this kind of mice here. So here we have the lungs of of mice injected with control cells, and we see metastasis in the lungs. But here, when we downregulate again cyclin D1, we don't see any metastasis. Here we have an hematoxylineosine staining that allows us to see cells really easy. Okay, so here we can see the cancer, here the cancer, and here there is not metastasis. But again, when we make paxillin hyperactive, we recover metastasis in the lungs. Without cyclin, but with paxillin hyperactive, we have lots of metastasis. And here you have the quantification, metastasis per square millimeter. So control has 4 metastases. Without or with low levels of cyclin D1, we don't have any metastasis, but when we do an hyperactive form of paxillin, we recover this metastasis. So what we can conclude about that? We can conclude that paxillin phosphorylation is required for cyclin D1 dependent cell detachment and invasion. Also, we can say that downregulation of cyclin D1 can inhibit metastasis unless paxillin is hyperactive, and that cyclin D1 and paxillin could be new targets to inhibit cancer metastasis. So now we are working on this part, trying to prove new pharmas or whatever that can inhibit any metastasis. So thank you very much for your attention.